So I'm going to just tell you a little bit about the Science Gateways Community Institute and what we do. So we provide services for gateways in three main areas. And the first area is providing support for building and running gateways. And that includes hands-on development support. Some of our team is actually on this call. We also provide advice from consultants on a short-term basis, so helping with things like cybersecurity or usability. And then we also have a catalog of gateways and software, which if you haven't a gateway or a component, I highly encourage you to go check that out and add yourself to the list. We look at it as being a way for people to learn about the resources available so that they don't necessarily have to reinvent technologies in order to make their gateway happen. And also, of course, other people can find out about using your gateway or using it as a model. We also provide a variety of education and training opportunities, which includes this webinar series. We also have what is called Gateway Focus Week, which was formerly called Boot Camp. And the Focus Week is, as it sounds, a week-long but very intensive opportunity to spend time working on your gateway, thinking through various aspects of how to make it sustainable and reach the audience that you want to reach. We have also, throughout the year, various student-focused programs, including a summer internship and a coding institute. Our annual conference just completed at the end of September, that is a great opportunity to meet with other people, and our website has resources and materials. And then our third area that we provide support is just in this opportunity to network and build a community. We do have a number of partners who provide complementary services and or software that you may want to check out on our website. And our affiliates program is related to our catalog in that it is for software vendors or providers who are willing to support the technology that they create for the people in our community who want to use it. So if you want to join the affiliates program when listing your software in the catalog, that is an extra opportunity to get more attention for your work. And then, of course, our website includes all sorts of news and job postings, and our blog. We welcome guest blog posts. In fact, Globus, which is I guess you could say the parent organization of the presentation, or the parent technology of the technology you're going to learn about today, they did a guest blog post for us recently. And then also, as you might have seen on the first slide, we have a community forum, which is a discussion group on Google Groups that you can join, and it's a great way to post questions and hear back from other people about things that you're thinking about. So those three are all complementary, and you can find out more about all of them on our website, sciencegateways.org. And then before I hand this off to our speaker, I just want to ask one quick favor, which is if you can take 30 seconds at the end of this webinar, please do give us your feedback about how helpful it was for you. And even if you don't feel like telling us that, we also have a space on that form for telling us ideas that you have for other topics that you'd like us to feature. We do actually look at that information and use it to help plan future events. So for example, this one is in response to knowing that people like to learn more about data and dealing with that. So I'll be posting this link in the chat area of the of Blue Jeans as well as we get partway through the talk. So with that, I'm going to hand this off to our speaker today, Ravi Madduri, who is affiliated with a number of different organizations. I'll let him explain all of his different affiliations and talk more about that. But he's going to be talking about the FAIR framework and how it is applied to the big data area, specifically in genomics. So Ravi, just so you know, I'm seeing right now your slides, but real small. So I think you have a really giant monitor. But when you run them, I'm assuming it'll, yep, that looks great.
Okay.
Fantastic, thanks.
Great. Can you hear me okay?
Yes, you sound perfect.
Okay, great. First of all, thank you for the Gateways community for inviting me to give this webinar. As Catherine said, that I do have a few affiliations. I'm a computer scientist at Argonne National Laboratory, and also have a joint appointment at the University of Chicago. I primarily work out of Argonne, and I'm a member of the larger Globus team, and this presentation came mostly from discussions with the Gateway community leaders on the importance of reproducibility, and wanted to kind of have the community be informed of the work that we've done in the last year and the year before on reproducibility in big data, and how that has contributed to important features in two products. One is, sorry, I'm trying to minimize this. How the work in the area of reproducible big data science have contributed and to features in two different separate but complementary products. One in Globus, in the data management and security management capabilities that Globus provides, and the platform capabilities from in the Globus product, and also in the Globus Genomics product, which is a vertical that is focused on doing genomics data analysis at scale using cloud resources. So I'll just, I know you all, and thank you all for tuning in to listen to this presentation. So let me just see if I can. Okay, so the work that I'm presenting today was made by a lot of work that's been done by the folks in this slide, and it's a result of a lot of leadership from Ian and Carl and Nathan. It's distributed across multiple groups, labs, from University of Chicago to University of Southern California to Institute for Systems Biology in Seattle. One of the reasons why as you could see when I present the work, it was doing reproducible research on a smaller scale is reasonably easier. But one of the things that we attempted to do, and we were quite successful in doing it, is how do we do reproducible research when the data sets are on terabytes, and the analysis that you would have to do is multiple million core hours. So that's one of the big reasons why this work had a lot of people who contributed to it, and I wanted to by this slide kind of personally acknowledge all of the hard work that folks in this slide have done in order to make this work possible. So this is probably the reproducibility crisis in science has gotten a lot more traction in biology and life sciences recently. And I think this is a general challenge in science where folks across multiple domains have tried to reproduce experiments, and how much success they have received in reproducing other experiments done by others versus experiments done by themselves. So one of the interesting, and one of the depressing study recently done by The Economist is to try to reproduce a bunch of landmark studies. Landmark studies are comprehensive studies sponsored by NIH and pharmaceutical companies that are essentially kind of a kind of a big deal in life sciences to understand or to essentially say that this particular genetic interaction is the reason for this kind of disease. So landmark studies are kind of quite a big deal in drug discovery and other important discoveries in life sciences. So a group, Amgen, try to reproduce some of the landmark studies in cancer research, and were able to only reproduce like 6 out of 53 among the landmark studies in cancer, and there are a lot of interesting data points in this article that The Economist has published on how there are so many studies, so many papers, so much science that's largely that's not been reproducible, and if you look at there's something called Retraction Watch. It's a website that you could subscribe to and see how many papers and how many publications have been retracted. One of the interesting things that's happened quite recently is the understanding of a genetic variant and its role in longevity of a human life, and they realized that after publishing it in Nature Medicine, this particular protein or genetic variant has received a lot of press in terms of its applicability in increasing the longevity of a human being, and then later found out that they made a statistical error in the genetic analysis and had to retract that study. So, and one of the things that researchers who've been following that have said was the paper was published without any reproducibility or any analysis or any study in the reproducibility of that research. So as you can see, reproducibility and being able to reproduce analysis and results that has been published is kind of a big deal, and it is important for how we can stand on others on giant shoulders to do research. So if the shoulders are not strong, then our foundations on which we are making discoveries are not strong. So one of the ways to make these foundations, these shoulders that we stand on stronger, is by ensuring reproducibility in research. So one of the important so we work a lot with Carol Goebel and others from in the UK who have done a lot of work on research objects and reproducible research. And the anecdote is that reproducibility is like brushing your teeth. Once you learn it, it becomes an habit. This anecdote comes from a realization that reproducibility is not easy, and it's like a habit that you kind of it's a discipline. It requires discipline that you have to adopt, and it's like brushing your teeth. And that's a picture of my daughter when she was younger, and it's a picture when we are trying to get her to brush her teeth, or trying to train her to kind of brushing her teeth as a habit. So it's not an easy thing. As you can see, she's not very happy brushing the teeth and gums that she had at that point. So at some level, like everything, it requires discipline, and it requires more of an elaborate and a thoughtful process. It's not magic. It doesn't just happen, but it's an important thing to cultivate and to include in your research. So we fortunately had a framework that we were able to look at. This is a paper that was published in PLOS Computational Biology by Anton and James and Sanvi, working together on what are some of the if you were to build a framework for reproducible computational research. and what are some of the rules that one could follow that could enable reproducible research. So these are 10 rules, and one of the distinction that I would like to point out is that we are here focused on the reproducible computational research. And one of the reasons why I'm emphasizing the computational part of it is because if you look at, especially in life sciences or in any other experimental research, there are strict SOPs or standard operating procedures in the wet lab or in life sciences that enforce a set of conditions or rules on how researchers would do research in a wet lab or in life sciences. In computational world, it's gotten a little bit harder to keep track of to enforce a standard operational procedure. So James and others in the study have outlined a set of rules that one could follow that could serve as a framework that could be used to generate results that could be reproducible. So these rules go from some commonsensical approaches like for every result in your paper, you keep track of how the results was produced. Avoid manually manipulating data. It's rule number 2, which is a common, it's a commonsensical thing, but it's a common pitfall that people fall into. Essentially, I'll just run this shell script, or I'll just do some manipulation on shell. These are the things that are oftentimes harder to reproduce. And archiving the exact versions of all of the programs that were used, controlling the versions of all of if you have to create custom scripts, how would you version control them? How would you record all the intermediate results? And these are all, I'm not going to read all the 10 rules, but essentially these are sort of it's a framework that one could follow if you're doing research using computational resources that you could follow to make your research reproducible. And in our in the work that we've done with the Institute for Systems Biology, with the University of Southern California, we use these 10 rules to make the research that we've done reproducible. And I'll tell you up front, it was one of the hardest things that I have done. So it wasn't easy to do this, but hopefully with a framework like this, you could use it. And one of the advantages that we had, in addition to this framework, a theoretical framework of the 10 rules, we also had NIH Data Commons, which was a system architecture that NIH that we were involved in creating in our joint work with NIH, which is sort of a set of software elements that would allow you to do reproducible research with the available data sets and the computational platform. So our approach in making data, in making research reproducible, is to adopt the FAIR framework, which is making the data findable, accessible, interoperable, and reusable, and use that framework to implement what we call as continuous fairness. We realize that making your data and analysis fair, it's not a one-time thing that you would do at the end of your analysis or your research. It is something that you would have to do continuously, making all the data that you're using findable, accessible, interoperable, and reusable at every stage of your experiment. And we wanted to use a identifier and exchange format conventions to implement this fairness. And we built on top of proven security and data and computational building blocks that have large user communities inside and outside biomedicine. One of the reasons why we did this is we also had a sustainability in mind. If we were to create something, what are some of the ways that we could make this sustainable? This is a big problem in life sciences. For example, if an instrument provided a data format and the company that made that went away, suddenly that data format is not reusable by anybody because it's not. So sustainability of infrastructure, sustainability of software, sustainability of data formats and computation is an important part of how we went about implementing the continuous fairness and reproducibility in the research that I'm talking about today. We also wanted to make sure that whatever solutions that we built them on top of existing best practices in the industry, so they have better chance of being sustainable and also are scalable and reliable, so that researchers can depend on them to do their science. So a lot of effort went into making sure that we do this in a way that by implementing processes that are sustainable and not like a one-off thing or one shot, so that it disappears after we have some success. So we built on some of the important capabilities of the Globus platform, mostly because, as I mentioned, because we want this effort to be sustainable. And Globus, we leveraged Globus Auth to make it easy for, as you've seen in my earlier slides, the collaboration included researchers from different organizations, from ISB, from University of Chicago, University of Southern California. So we had to make sure that all of the researchers, so we provided a way for them to securely authorize and authenticate to the resources that are being used, that are data sets that are being used in doing the science. So we used Globus Auth to implement the multi-institutional authentication authorization, and to be able to implement fine-grained security to enforce access to data sources and be confident that the right people have access to the right data. And we built on top of the Globus data management platform that made it easy for us to use cloud S3 buckets. So all of the data we used in the study that I'm going to talk about, all of the data was on S3 buckets or on web data sets that are available through web interface. So we used the Globus servers to make S3 buckets be available through a Globus interface. And one of the reasons why our life was made easier is because we had a uniform form protocol to access data. So all of the data objects that we're using in this experiment or in this study were accessible by one access mechanism with Globus. And we were able to use multiple compliant authentication protocols or authorization protocols for us to access the data. So this is an important part. This is probably the part that folks in this community may not have heard too much about. But we enforced interoperability by creating two capabilities. One in the way we name entities. We named these digital objects using unique identifiers. We came up with an identifier framework called MinID. And people who work in this domain could already point out that there are a lot of identifiers frameworks already available, like DOIS and Handle, or some of them. What we've done we haven't created yet another identifier framework. We created something that would make it easy to add an identifier and some metadata to that identifier, so that these data sets could be identified or could be discovered in a registry. So MinID stands for minimal identifier, essentially combines an identifier with a checksum so that we can vouch for the integrity of the data. We also used existing identifier protocols like a handle or DOIS, and data site is another one that we used to uniquely identify data objects. And we came up with a format for exchange called beady bag, which stands for big data bag. And again, here we haven't really created it in vacuum. We extended an existing W3 standard called Baget, which is used by digital libraries to uniquely identify digital objects. So we used Baget specification to create a beady bag to describe a data format that will make it easy to exchange large data objects easily. So that with MinIDs and with beady bags combined, we were able to describe digital objects that could be easily shareable and between multiple parties with the other checks like integrity checks built into the protocol, so that when I share with you a beady bag with the genomics data, you know that because the specification includes checksums for the files, you know that the integrity of the data is preserved when you exchange them. So as part of this, we created Workspaces that makes it easy for this data to be computed on. So Galaxy and Globus Genomics is one such workspace where we created capabilities to make use of beady bags and identifiers to and track the provenance of the analysis and be able to run this analysis on various infrastructure elements like Amazon and Google Cloud by enforcing cost-aware resource allocation for both not only for computing, where we leverage a lot of spot instances on Amazon to keep the computing costs low, but also data movement. So one of the important attributes that the beady bag and MinID and identifiers will give you is the notion of data locality. So you know that this particular identifier resolves to data that is located on a cloud on Amazon, so you would want to run the compute on Amazon so that you're not paying for moving the data to another cloud or to a local storage. So a lot of the data-aware, cost-aware, data locality-aware compute is baked into these workspaces, so you could enforce some of these compute. So we implemented jointly working with University of Southern California. We created a resource, a registry where you could discover these digital objects using the metadata. So for example, if I'm trying to discover a particular set of data that match a metadata element, like I want to analyze data sets that are of human beings, of kidney, then Deriva allows one to create or search through the digital objects using the metadata. So what we've done in this work is essentially bring all of these capabilities together to create a platform that will enable continuous fairness of digital objects. So we integrated with publicly available digital object resources in the NIH space to make it easy to discover these digital objects using their metadata, be able to create beady bags of the digital objects that you would like to analyze, and mint identifiers for those digital objects, and use the analysis platforms like Globus Genomics to take these digital objects and run large-scale analysis while keeping track of the tools and the intermediate data sets that were generated from doing this analysis, and all the complex computation that you would use to do this analysis, record the provenance of that, and record that in such a way that you could publish it and somebody else can reproduce the analysis that you've done and take a look at the data that you have generated so that they have more confidence on the results that you have generated. So this is rather a busy slide, but it kind of uses all of the different components of the platform that was used to enable this case study that I'm going to talk about. So using this platform that enables continuous fairness, we were able to create an atlas of transcriptional factor binding sites from the ENCODE data set. So ENCODE is a publicly available data repository maintained by NIH that gives more information on functional elements of human genome. So our research included understanding what are the putative transcriptional factor binding sites that are already available in ENCODE, and use this transcriptional factor binding sites to better inform drug discovery for diseases like Alzheimer's and traumatic brain injury and other neurodegenerative disorders. So we wanted to use that as a driving science question that will allow us to do a lot of analysis on using the ENCODE data set. So we have two preprints on this work. One preprint talks about the science, and I put the URLs and DOIs for them. The science that we are trying to enable using this approach. The second one is the infrastructure, the computational framework and the platform we built to enable that science. So I'm happy to tell you that the infrastructure paper, the reproducible data science paper, was accepted at PLOS 1. It's now available, and the science paper was submitted it's under review at Cell Systems. So we'll probably hear about that very soon as well. So I'm telling you, so my goal here today is to tell you the infrastructure that was used to enable this cutting-edge science. And how we are able to make this the science question be answered in a way that it is reproducible end to end. So this slide so the left-hand side talks about the workflow that we had to implement to analyze this data. So it involved a lot of steps, a lot of analysis steps, a lot of data management steps where we went to the publicly available ENCODE data catalog to extract the data sets and then create beady bags of raw data. And then we used Globus Genomics to do large-scale analysis on this data to generate alignment files, and then used an ensemble of footprinting algorithms to further analyze the alignments from the ENCODE data to generate this transcription factor binding sites. And then we've done a lot of statistical analysis on these binding sites to know whether they're putative or not. So on the right-hand side, you would see these are the tables that we listed in the paper, in the publication. As you can see, they're identifiers. These are web resolvable identifiers that take you to the data sets that were generated as part of this study. And on the bottom, you'll see identifiers for all of the analysis tools that were used to generate the data sets in the top table. So we created a easy to use interface that would help create beady bags and identifiers for the raw data, and then once we have the beady bag for the raw data from ENCODE, we created a comprehensive workflow in Globus Genomics that takes the beady bag as input, resolves the identifier, finds where the data is, and the data happens to be on an S3 bucket powered by Globus endpoint. And it uses Globus transfer to get the data, uses Amazon Spot instances to run this analysis at scale, generates a whole lot of outputs, and then generates a beady bag of outputs and identifier for all of the intermediate data sets and the results, and then puts them back into an S3 bucket for further analysis. So this is how this is the breakdown on the data. On the ENCODE, we analyzed 193 samples across 27 tissues, and it took, this took a lot of core hours. These are to do this analysis, and we've done it a few times using different algorithms so that we could get a very good, powerful data set that others can do more science on. So we also ended up creating a GitHub repo that outlined all of the steps that somebody could follow to reproduce the science or reproduce the results. And we included instructions on reproducing a small set of data, because one of the challenges that we had was reproducing the entire study roughly means you would have to run around 100,000 CPU hours on AWS. So the sustainability and the cost of who would bear the cost is something that the economics of reproducibility is something that's an issue that should be addressed. Some of the lessons learned during this process is that we realized that reproducing a large-scale data analysis or creating a data analysis pipeline that you want it to be reproducible requires a lot of patience and discipline. I say patience because we've had many. We had to redo a lot of analysis because we weren't careful when we ran some of the data sets initially, and we had to make sure that researchers in a distributed team don't just open up a bash shell or just write a Python script to do some data analysis. So we had to capture every step of the analysis. So the complexity and level of effort that is required increases with the size of the team that is working on the analysis and the number of data sources. And the life cycle of the data, and the economics of who's going to pay for archiving, who's going to pay for long-term storage of this data and the analysis results is still an open question. And the sustainability of the infrastructure, some of the infrastructure that we built and used is also an important question. So so I wanted to kind of end with acknowledgements from the Galaxy and Globus team for working with us and helping us in making this possible, and a whole lot of funding from NIH and DOE and NSF for building software elements that we use to do this work. So that's pretty much what I had, and happy to take questions at this point.
We do have a question that just came in. Thanks, Mona. She said, can a science gateway who offers computational resources but not long-term storage of users' data use or add your framework to offer some of the needed information to the user for reproducibility?
Yeah, that's no, definitely. So some of the key capabilities that we built, like the beady bags and the identifiers and the Galaxy tools that would resolve beady bags, all of them are openly available. So so some of those things could be applied and adopted in your own science gateway to ensure or to kind of make some of the analysis be reproducible.
Great, thanks. Waiting to see if there are any other questions coming through, but in the meantime, I was just curious. So Genomics is one. Sorry, she has a follow-up question, and I'll save mine for later. She said, do we need to add the code to our gateway, or can we call your REST API?
So that's again a good question. So I think there are different components here, right? So there are components for data transfer, components for creating beady bags and uh, generating identifiers. So all of the Globus related data transfer components, authentication, they all have very comprehensive REST API. For the beady bag component, there's openly available GitHub repo that you would have to it's a Python library that you would have to use that code to generate the API. So it's a mixed match, I guess. There are some of the components have APIs, like Globus Genomics. We have REST API that you could use to call, and some of the components you would have to download and use their Python libraries to integrate that into the gateway.
She said, okay, thank you. All right. So while we wait for another question, my question for you is, so I understand that genomics data is a particularly ginormous and, it's a very big data thing and complex and so forth. So how do you see this translating to other kinds of data? How hard would it be for someone to adapt? In a way that's a follow-on to what Mona was asking.
The only science-specific component that I presented today is the registry or data catalog, where you would have to have the metadata be loaded for you to do the discovery. But most of the other components are extensible and useful for other domains. For example, let's just take Globus Genomics and Galaxy as an example. So, we build Globus Genomics using Galaxy and Globus, but we also use Galaxy for Cosmology. So PDAX is a gateway that I'm involved in that uses Galaxy to do cosmological simulation. So all of the tools that we develop for reproducibility, for managing identifiers and beady bags in Galaxy could be applied to gateways that are Galaxy based, I guess. And there's not a lot of science specific components here. I think one could reuse them across multiple scientific domains.
That's interesting. That explains why I saw my colleague Gus on the on the list of attendees. So I was wondering what cosmology had to do with genomics, but now I know.
Actually, that's news to me that there's a cosmological use for this. I'm very happy to see that.
That's great. Well, I'm glad this could be useful for you, Gus. I'm happy to see you here on the call. Okay, so I'm still waiting to see if there are any other questions coming through. That was pretty comprehensive, so I have to say I'm not surprised that we don't have a whole lot of puzzlement. Oh, is there someone who wants to come in with a question? I heard some noise there,
Yeah,
maybe.
Yeah, I thought that was, I think I thought I killed Outlook, but it keeps beeping. So.
Oh, that was that was just your own computer. Oh, okay. It sounded just like a person wanting to ask a question.
Yeah, they're getting good.
Yeah. So, all right. Well, I'm still waiting to see if there are any other questions. I will ask you another question, which is, you said that you were in the beginning of your talk, you were saying how tackling reproducibility has been one of the most difficult things that you've had to do work-wise. At least it sounded like, it was equivalent to me if I were trying to start an exercise program and like run every morning for 5 miles. That would be really hard for me and a little bit miserable and so forth. So I'm wondering if you have advice about how, jumping into that kind of thing. Obviously this framework is also an opportunity for people to, it's meant to make it easier for them, that process. But what else is there about the whole reproducibility goal or the fairness goals that that you've learned from your experience that might make it easier for folks?
Yeah. So one important distinction that I like to make from your, I mean, from running a marathon or learning, I mean, starting to run 5 miles a day. It's things are a little easier if it's just you and you could. But science is it's a team sport, right? So um, in our experience, if there are multiple teams with multiple capabilities working together on doing something big, chances are that there's going to be it's the discipline and the making sure that everybody understands that everything has to be recorded, everything has to be published so that somebody else can reproduce it. It requires a lot of inherent discipline and a lot of attention to detail. And luckily for us, we had Ian Foster
Mm-hmm.
be the overall. I mean, he was kind of the guiding. Essentially, he was making sure that we record everything. And Ian has a very gifted sense of tracking details. I think he's really good at it. So
Uh-huh.
so he was having him on the team helped us get better. And I would it was pretty. It was not an easy thing to do, but especially when you have multiple teams working together. So the framework is only as good as the discipline and the want of folks to make their research reproducible. So I think that they both have to be there. I think people would really want to have want to do this in a way that it's reproducible and have the tools at their disposal to do it without having them to create these tools.
Great. Yeah, yeah, I see how that would be. And it sounds like it's really, in a way, something it's a good idea to start instilling in graduate students very early on, because it's not I mean, I know that as a grad student, I used to find it amazing that people couldn't, remember things about their own research a year before until I started doing research and realizing I couldn't remember what I'd done a year before. So
Yeah.
does take a lot of discipline. So we have several questions that just came through. Marcus asks, do you keep BD bags of intermediate results in the workflow? And if so, does this allow reproducing part of the analysis?
We do. As you know, or probably already know, so Galaxy does keep track of all of the tools and the parameters that were used to run the tools. And we used S3 to archive the intermediate data sets and eventually moved them to Glacier because they were getting quite expensive to maintain. And some of the intermediate data that we that didn't involve stochasticity. One of the determinations that we made was that if I could reproduce the result without without loss of information or change of information, then it's probably okay to just keep the version of the tool and the input to that tool so that we just record it so that we know how to reproduce that data. But when we ran the experiment, we stored all of the BD bags of all of the intermediate data sets and in S3. And the second question that you had was the long-term archive of these identifiers and data sets when in 100 years, when BD bag and MinID isn't there, how does one reproduce the workflow? That's a great question. I don't know the answer. I mean, that's the problem that I want to work on. That's the question that I want to work on. I don't know the answer, and I've thought about it a bit, and I don't have any good insights into it other than let's target for 5 years. Then 10 than 20. I think even in the 5 years, at least in genomics area, the data standards have been reasonably stabilized, like FASTQs and BAMs and variants, and the tools that. So it's a whole ecosystem, and it's the whole economics of it. It's not it's a non-trivial thing, and it's a hard problem, and I would like to work on it. The other question is from David. How aligned is your platform with GA4GH specification? So it is pretty, I mean, it is reasonably aligned, I guess. So we use WES. If you're familiar with GA4GH, or GA4GH is it's a genomics alliance for, no, it's a global alliance for genomics and health. And I'm a member of it, and I'm going to the plenary in next couple of weeks from now. So we use some of the interfaces that they have generated as standard for execution of workflows. We use CWL in our platform. We use some of their standards in the data, like FASTQs and VCFs that adhere to GA4GH specifications. But I think there's a lot more work to be done, because there's a lot more specifications and standards that they implement. I think the last question is, do you know if NSF, I mean, not last, but last in the chat.
Latest, yeah,
Yes.
from Mona.
Do you know if NSF, NIH, and other grant agencies support and enforce reproducibility? That's I mean, I know NIH is investing or has grants in are asking for fair metrics for data. So I know NSF too, but I don't know off the top of my head. That's something that I think everybody says that they want to do reproducible research, but as I said, it's the economics of it are still not very well understood, and that's, I think there's active work going on in funding agencies to get their heads wrapped around all of the challenges that are involved in using enabling reproducible research.
Yeah, and I would add to that my other sense that at least I've heard in complaints about the way agencies fund is that they're always, one of the problems is they're often looking to fund new stuff, and they want, the new and then innovative and so forth. And if you go to them and say, well, hey, I'd like you to fund me to do something that somebody else has already done just to see whether it matches, they're not as keen on that typically, and that's a real problem inherent in some of the funding pathways. Even though obviously this has been a huge campaign on the part of scholars who recognize the importance of reproducibility. I mean, that it's acknowledged as an academic concern, but it's not necessarily acknowledged in the way the direction of those funds and the mission of those agencies have been conceived, I think. Yeah. So I don't see any new questions coming in at the moment, and so I'll take a moment just to take the opportunity to mention first that I'm very grateful for having you here today, Ravi. I appreciate you pulling this together and sharing this information with us, because I think it will be valuable for members of our community. So thank you for that. I certainly learned some good stuff today. I appreciate the thank yous also coming from the audience members. I'm glad you guys have enjoyed this. So thank you, and we look forward to having you join us next month.
All right, great. Thanks a lot, everybody.